Okay, magandang hapon sa inyong lahat. Thank you for attending this seminar, one of the series for MSI 50th. So, I have the pleasure of introducing our speaker today. Um, Aletta is a graduate of BS Biology from uh, Los Baños, magna cum laude. Valedictorian, alam nyo valedictorian. And then she went on to do her PhD at the Rosenthal School of Marine and Atmospheric Science in the University of Miami. And then came back to MSI. Pero sabi nga ni Alet, right after right after graduation, her first job and only job to now is still with the Marine Science Institute. So she's one of our homegrown. She started as an RA and then went on to do her studies and came back. Okay, and now um, she's a professor and runs the biological oceanography and modeling ecosystems, or the biome lab. Uh, her scholarship was from Fulbright and Maytag. No? So she specializes on marine ecosystem dynamics, developing integrated models and decision support systems to address harmful algal blooms that enhances conservation efforts in fisheries management. She is also very well recognized with the L'Oreal UNESCO for Women in Science Fellowship. She's also an outstanding young scientist, and more importantly, currently serves on the scientific steering committees of several regional and global initiatives, no? Including Decade of the Ocean. She's also the Philippine representative now for the IOC Westpac. So uh, I have the pleasure to welcome and introduce Alet for her talk, The View from the Bottom Up. Alet. Thank you, Annette. Yes, so yeah, my very first job after college was here, MSI, and left for my studies and still came back. So, and more than, oh my gosh, 15, 16 years na since I came back. Huwag na tayo mag-math. um, so, actually, what also today, it's a mix. It's a bit of going back into the roots of how I started here. Looking at ecosystems from what I term as the bottom up, and we'll get to know what that actually means more. So first, ooh, of, of course it's not working now. It was working earlier. Hang on. There. So when we talk about ecosystems, so the common ways to think about and represent ecosystems, there's usually two main ways. The first is thinking about it in terms of the trophic web. The focus is on the trophic levels. So you have, of course, your primary producers to your herbivores, your first order consumers, and so on to the top predators. So the focus really is on prey and predation aspects, right? Now, the other perspective, but of course, it is really in essence, it's still trophic pathway based, but the kind of the nuanced thinking about it, it's more on cycles, not necessarily just prey and predation, but you have the cycling of your elements, whether it's carbon or nitrogen or phosphorus or other
elements that you would prefer within the ecosystem. So, and it's not necessarily just through prey and predation, but it can also be through other interactions. So that would be, for example, in this second figure here, your representation of the biological carbon pump, where you have the carbon cycling through different parts of the ecosystem, whether it's your also your plankton or to the higher trophic levels, but also not just through predation, but the other interactions present there. And then what happens to that carbon as well, whether it goes back to the atmosphere from the ocean or settles down and sinks and is sequestered into the seafloor. So those are perspectives about the ecosystems. Now, usually, there's actually kind of a two different perspectives when you view and represent ecosystems. So that first one, it's more process oriented or top down, meaning you think of the individual organisms separately. You bulk them up, and for example, you have your prey population or your prey community as a bulk, and then the transfers to the next part of the ecosystem, like your larger predators, is one flow from one part of the system to the other, and you designate the rates of that flow. Right? So you then consider specific individuals and their diversities, and the rates for these processes are prescribed and usually averaged out. Um, as I'll give more examples later. The other perspective is what we call individual based approach or the bottom up approach. And here, the consideration is that your ecosystems are made up of course, you have your species and your different trophic levels, but within those groups, you have individuals that vary and interact in a more local manner. And that interaction in a localized manner and that variety between individuals, you allow them to play out, and there's high level rates, whether it's population rates, growth rates, or community respiration production rates. Those rates and processes come out or emerge from the individual's actions and localized interactions. So just some representative examples, your classic models, whether you present them as, for example, differential equation models and food web models, you have these compartments. So trophic level 1, 2, 3, 4, you have these compartments. So you don't need to read these, but for example, some of them are detritus, primary producers, or the consumers. And then these lines just represent rates of flow from one compartment to the other. And it's kind of a more top-down approach where, okay, we have your compartments made up of this bulk set of organisms, and then the flow from one compartment to another is something that you set, right? Of course, based on different studies and what's known. The other, for example, we've actually also developed, apart from bottom up, I do times do top down approaches. So in the past, we've done the Filipinos nutrient, phytoplankton, zooplankton, detritus model, or NPZ, present these bulk groups. For example, here, diatoms, phytoplankton, zooplankton, and their different nutrient types into boxes, and then again, prescribe the flow from one compartment to the other. But we don't have individuals. We just say, okay, in this cell, there's a concentration of this amount or biomass of this amount for this group here, for one of those boxes here. All right. So that's the more top-down approach. And usually that's done through your numerical models or differential equation model approaches. Now, the bottom-up approach, this is something that I was exposed to from my PhD. It really fascinated me that this kind of framework. So it comes from this complex adaptive systems perspective where the whole is greater than the sum of its parts. Okay? So complex adaptive systems consist of diverse interacting agents. So we use agents because sometimes an agent doesn't have to be an individual, and I'll explain that in a bit later. So consists of diverse interacting agents that adapt or learn while they interact, and that organized behavior emerges from the interactions without any global plan. So there's no leader there, but there's within that system there's self-organization, capacity for self-organization. And what is really interesting for me with this kind of perspective is you can really have an analogy across disciplines. It cuts across disciplines. So whether you talk about an individual in a population or species in an ecosystem, businesses, cultures, languages, neurons, branching networks of food webs or rivers, you can have this perspective that has a similar kind of patterns and processes in play. So, for example, you can think of an economy with firm 1, firm 2, firm 3 as the agents, or an immune system with antibody type 1, 2, 3, or a nervous system neuron 1, 2, 3 as the agents, and their local interactions and the differences between them um, interact so that they produce this emergent higher level property. So for economy, for example, it's a gross domestic product, GDP, or immune system, an individual identity, or for nervous system, a behavior, or for your individuals in the population, you have your connectivity characteristics, or your different your ecosystem characteristics, and so on. So just as to further highlight this concept, so when you think of it, have of course, at least some of you have actually directly seen schools of fishes. So who has who has directly physically seen schools of fishes? Raise your hand. Yeah. So have you ever thought and wondered how do they school? Is that I mean, it's a bit of a maybe I don't know if it's a philosophical. I think a legit scientific question. Is there a fish leader who says you here let's form a ball, a circle, a line or something, right? So actually that seems very anthropocentric, I don't know, anthropomorphic. But actually, schools can emerge based on individual decisions of the fish. So there doesn't have to be a fish god or fish president, you know. Each fish, based on its localized space and interactions, that can actually by themselves, lead to the schooling of the fish. So what has been found in other studies is, for example, three navigation rules only can allow schooling to emerge. So these three, for example, rules are cohesion, alignment, and separation. When you say cohesion, it means if I'm a fish in a group of fish, I don't want to be too far from from the other fish, so I will try to move closer. Alignment is based on what's going on around me and the other fish. I will try to kind of generally align myself in the same direction. And then separation is I don't want to be close either, that I'll bump into them or whatever. Then I will a certain distance between myself and the other fish. So those three rules, if you let them play out, those actually lead to fish schooling behavior. And some actual scientists have actually also measured these kinds of um, metrics in fish schools and saw that okay they are they have basis. And so this also applies not to fish schooling but also bird flocking, right? Same similar kind of formations. So for me, when I was looking into these things, because my advisor then was, he's very interested in this kind of approach. For me, I was like, wow, this is really amazing. I hadn't really, in ecology, my ecology courses in undergrad, I hadn't really, these things are part of the curriculum. So these were completely new at that time. But now it's more, I think, there's a lot more studies using this kind of approach. So applying now to more on the marine ecosystem side. So viewing marine ecosystems as complex adaptive systems. Of course, we contend with there's diversity in the host species and even within species in the systems that we're studying. And there's definitely a lot of spatial heterogeneity. So if you're working with coral reefs, we know there's a lot of patchiness that you can find across the bentos. At the same time, in the pelagic environment, even in a small space, there can actually be a lot of patchiness at the micro scale. And you have your interactions. So on a small scale or individual scale, actually these can emerge and cascade to the larger scale patterns and processes. So like I mentioned, you have, for example, the individual dispersion interaction of your larvae with its ecosystem, and then but then again you have your larger connectivity patterns emerging from those. And of course, more classical models show more average conditions. And so we definitely that we need to consider the variability in conditions, and there's a chance of change, whether it's more of an immediate short-term change or long-term evolutionary change for your organisms. And that that's difficult to represent. That actually needs to be considered, especially when we're talking about long-term variability or long-term changes like your climate change. So here I'll just highlight a few examples of this approach, and probably some of you have seen this, but I'm going to delve deeper into some of the aspects behind the models. So two applications, a sardine population model, and then one is on the HABs or application. So the way we represented these in models is that what's called agent-based modeling approach. And agent-based models is the same as individual-based models or multi-agent systems. So it depends on which part of the world it refers to the same method. And essentially it mirrors the complex adaptive system perspective, where you represent the behavior, the variability, and the interactions among the agents, and you allow for the emergence of higher level or sophisticated properties from the simulated agents. So for the PO ones, you're very familiar with this Lagrangian particle dispersal approach. It's an EBM or IBM because you're tracking a particle, an individual particle. But that's the most, in a way, simplified version of IBM because it's a simple particle where you're tracking it because of the circulation vectors. But on the other end of the spectrum, you have the more complicated agent-based models where you actually incorporate a lot of traits, behaviors, life histories, and especially even more complicated, the capacity to adapt and evolve in the because of different environmental conditions in the model. So for the sardines, we were focusing on the northern Zamboanga Peninsula. area. And so the concept is this is a simplified relative, life cycle of your sardines where they spawn, produce the eggs, and which transition into the larvae, and then you eventually recruit into the what's considered a stock that can be caught, your adult sardines. So we were interested in relating stock variability due to the recruitment variability. So the idea is that your environment, in this case, sea surface temperature or temperature and chlorophyll, which is a food indicator, and your ocean circulation can affect the sardine larvae. So the sardine larvae are actually they're omnivorous, but more biased towards your zooplankton. So here we didn't have a way to represent zooplankton explicitly, so your indicator more was the phytoplankton to your chlorophyll. And then so we were interested in looking at this pattern. So based on work done by Dr. De Guzman and team, um, they and other studies, they showed that Sardinella lemuru spawn during specific time periods in the year, from around November to February is the spawning period. So the so why is this? Why is there a particular time period within the year that's the spawning period? So some hypothesis for that is, of course, this would be the optimal for having a faster growth so that it would be able to escape, for example, mortality or predation through like predation, so that it can grow faster and get into the size that it is not as vulnerable compared to the smaller sizes. So we look more into this to assess what are the conditions during that time period when it's spawning, and then how that then affects the recruitment into or potential recruitment into the stock. So we used this model coupled we used a circulation model coupled with it's actually more simple particle model. So the sardine in this case does not have didn't represent it for this model the any complex life cycle aspect. It will, but we did track what kind of chlorophyll or potential food environment it encountered in the space, the circulation, and then of course the temperature. So we ran this simulation for every 5 days for 6 years. This was 2008, 2014. Tracked what it experienced, what each larvae experienced for 60 days. And we actually had 0 to surface to 30 meters, essentially tracks about more than 70 million particles in for all of that time period and what conditions it experienced. So yun. So so this is just a simulation. The green is the satellite-based chlorophyll, and then the red are the sardine larvae. So there's also a connectivity aspect here, but I won't really focus on that. So what were the environmental conditions experienced by the larvae spawn during the peak spawning periods compared to those other months of the year? Turns out during that spawning period, it experienced, interestingly, lower larval retention. So when I talk about retention, it's sorry, from this previous here. We say that it's been recruited into the stock if at the end of the tracking time it was the larvae were within this 15-kilometer, it's about 200-meter isobath range across the from the coastline. So what we saw that it had lower larval retention. So this was actually kind of surprising because, okay, well, that means during the spawning period, there's more of a tendency for the larvae to be transported offshore. Now, exactly where didn't really track anymore. So, but within that area, at least, it was not retained anymore. Right? And then, but what we would hypothesize is during that time frame, during the spawning peak spawning period time, there tended to be relatively higher chlorophyll conditions, so potential food, and lower temperature for the recruits. This was about 28 or so degrees Celsius, which is cool compared to the rest of the months of the year. So this conformed with um, what was found that Sardinella lemuro had about its the optimal temperature was 27.8 degrees actually. So I was thinking then, because this was at this time point, 2011 was the start when in a sardine fishing ban in the area. And the fishing ban at the time was December to February. Now it's November to January. And they instituted it for 3 years during this December to February. And of course, we know that, of course, your stock is influenced by fishing. That's a mortality source for your stock. But if you think about your ecosystem-based management perspective, you have to consider the environmental aspect. But so when we were looking at the catch at the time, 2011 was the start of the fishing ban, and then 2012, 2013, 2014, when the ban was in place, it looks like the catch is increasing. So was this a function of because the catch, because of the ban that was instituted, and so that helped to increase the stocks? And of course, in the news, that was what before was saying, right? Which is fine. But like I said, if you think about ecosystem based management, it has to be holistic. So yes, you have your fishing mortality and you're taking that away from the ban, but at the same time, you have to consider, well, what about the supply side? Was there any change in the supply side for the stocks so that the catch would increase or not? So that was what we wanted to check. And how did, because the one I showed you was on a within a year and a monthly basis. So how about interannual variability in terms of the conditions explaining the recruitment and potential stock variability? So during the high landings and fishing ban implementation years, the environmental conditions for recruitment actually improved. So this is per year now, 2008 to 2014. And it was this during the closed ban period. So during that time versus the previous non-fishing ban periods, there's actually higher potential for retention of the recruits. And then also over here, there wasn't that much of a difference in terms of chlorophyll. It was high, but it was in terms of the overall, the actual concentration, like a median of about 4 grams per cubic meter. That's actually considered pretty high in terms of chlorophyll concentration in general. And then the time when during the fishing ban period, it's actually closer to the optimal temperature that Sardinella lemuru preferred. So putting all of that together, like I said, yes, the fishing mortality probably decreased because of the fishing ban, but also on the other hand, your potential recruit supply could have increased as well because the conditions for the environment were more conducive to the survival and growth and recruitment of the larvae to the stock. So not to say that don't do the fishing ban. It's just it has to be a holistic picture when you consider these things. So we actually use the same model. So these models, the nice thing can be very reusable. So that same model we use now to assess larger, like 20-year worth of um, um, uh, like 20 years in the area from 2002 to 2022, so assess the impacts of ENSO and any fingerprint for climate change. And essentially, those patterns that I showed confirmed, but also now the influence of those larger scale changes from ENSO and climate change, we still have to tease that out. ENSO more so than climate change is also impacting those environmental conditions. So essentially for the sardine population, we saw that even if we only represented those larvae more in a simplistic way, but tracking them and the spatial heterogeneity and the localized experienced environment affected its potential recruitment to the stock. And so now how the how that environment changes with those ENSO and other climate change factors, it's a uh, I think important question now to assess further. So that's for usually now for the hubs. So for the hubs, we're actually going up a notch now into the gradient of more complex representation of the individuals. So hubs, I'm not gonna go to too much descriptions here, but I assume you have an idea what hubs are. So hubs, red tide, can be fish kills. It's the proliferation of algae causing direct and indirect negative impacts to aquatic ecosystems, coastal resources, and human health. And of course, as you probably know, issues with hubs affecting many benefits in the Philippines and different types of hubs. So there's toxins causing harm to animals and humans through toxic seafood consumption or waterborne toxins. We also have high biomass species that disrupt the ecosystem or food web or cause fish gill damage or low oxygen or anoxic conditions. So, Paidium bahamense, our favorite hab organism. It's a key, it's one of the main, it's really the main species causing a lot of toxic blooms in the Philippines, and they typically occur in embayments, in different embayments in the country where you have cyst beds of the pyridinium serving as seeds and then blooming at some point in time. Manila Bay used to have pyridinium, now it doesn't. The idea is because pyridinium doesn't like very eutrophicated waters. It has now been replaced by gymnodium and alexandrium, which apparently tolerate more polluted waters better, like Bolinao, Alexander. Anyhow, we still need to prove this, Irene. So, the come up with a holistic model for hubs. So we combine physical, chemical, geological information with the hub causing organisms and also the toxin vector, which is the shellfish. So in this study, we focused on Sorsogon Bay to understand factors and mechanisms leading to pyridinium bloom initiation, development, and decay in tropical embayment like Sorsogon. So, as I said, so now it's more complicated. This is now has more of a life history and behavior for the particles or the individuals. So your agents are your cells of the pyridinium and the cyst of the pyridinium. So we are representing the life history of pyridinium. You have a vegetative cell that undergoes growth or asexual reproduction in this case, encystment, excisment, the cyst stage, which can also undergo mortality. So both undergo mortality and also can be affected by the water movement. And these life history processes are affected by light, temperature, salinity, and your nitrogen. And then, so the other part of this model, of course, so you have the hydrodynamic model, and they also, in this case, the watershed model developed by Lau and Ko for the input of nitrogen into the bay. So this was coupled with another biological model, the shellfish representation. So we represented two compartments, internal, the tissue compartments as based on studies, and the transfer rate between these two parts of the muscle and then the toxification into the environment. So just as an example of local interaction, so each cell in the cell, for example, of pyridinium, would its growth is a function of those what I mentioned, light, temperature, salinity, nutrients, and based on its local space. So it's not average over a Sorsogon Bay, but in the cell it's occupying at this moment in time what are those environmental conditions, and then how it will grow is based on, of course, you still have equations. You can't avoid equations. These were based on different studies, past studies, the way we represented these. And the shellfish as well. So the shellfish actually was eating or filtering the cells and the cysts from the local space around it. So depended on the advection, depended on of the cells or cysts of pyridinium if it was able to filter it out. So again, this is the representation of the model. Green are the cells, blue are the cysts. So you have cyst beds based on Dr. Serena and Co.'s information on the, we had huge, a lot of samples of sediment to look at cyst beds. And then mussels are the white dots by the coast. If it's red, it means it became toxic, meaning it went above a certain concentration in its tissue. So, yeah, so it's just fun to look at simulations. Anyway, so eventually it's later when usually the blooms are more the cells and cysts are more concentrated inside, then you start seeing the uh, toxic muscles. Saan ba? Sorry. Ang hirap kasi delete nung ano, yung pula-pula na. So that's when usually you have the shellfish, toxic shellfish occurring at the end of those more southwest monsoon. So things that we learned because of this approach, we were how do I say this? We did many versions of the model to assess which processes, which parameters were the most important, right? So some things we saw were various cell densities and shellfish toxicity were strongly significantly correlated to mortality of cells and cysts. So negative. So of course if there's a high mortality of the cells and cysts, then you have less accumulation of the cells and less toxicity for the shellfish, but also that cysts were important. So if there are more cysts available, then you tend to expect that there would be higher cell counts and also higher shellfish toxicity. So okay, that makes sense. Now the aspects that we looked at using the models. So here's a temporal pattern of pyridinium cell density and shellfish toxicity. The red one at the bottom, that's observed data from before. So time series data. And the rest of these are the different versions of the model that we looked at. So essentially, the main pattern that we saw, of course, is that mainly the blooms occur during the southwest monsoon, usually towards increased rainfall. Not all the model versions, by the way, fit. So we were looking at different versions to use that to help us figure out which processes were more important. But anyhow, the main pattern is that blooms occur during the southwest monsoon when rainfall started increasing. And so we wanted to look into that further. And because when you think of rainfall, um, there can be two ways that it affects HABs, main ways. One is how it affects the circulation. So it can create a more stratified environment that can lead to accumulation of the cells, physical accumulation of the cells, or it can increase runoff and thus the nutrients runoff as well into the belly, which can promote the growth of your pyridinium. So that how do we tease those apart? So we can actually use the model for that, and that's the fun part with the model, which normally you cannot do that in real life. You change the stratification or change the rainfall, right? So here we did scenarios in the model where there was strong, moderate, and weak stratification coupled with combinations of high nutrients or hypothesized expected nutrients where if there's high rainfall, there will be high nutrients, medium, low rainfall, medium, and low nutrients, respectively. And then we also looked into grazing. So what would be the impact of shellfish grazing on the cells and the cysts as well? So that's a kind of multifactorial experiment in the model. So what we saw in if we had high medium stratification, but no difference in nutrients, there wasn't that much of a there's still high pyridinium density across. But if you now put in the appropriate assumed nutrient input because of high, low, mid rainfall, then you have a large drop in the pyridinium cell density when you have low nutrients as well. And then with grazing, that would be an additional decrease in the cell densities of pyridinium for both scenarios. So it seems like based on all of these, putting all of that together, cis as an initial seed, very important. So maybe the more cis, there could be a higher potential for bloom. But existment does occur actually across different seasons, but then the cells can increase if there's the input now of your nutrients. And that rainfall can be the trigger for that. And the plumes are could be strongly affected by your mortality processes. So that's the things that we put together with that. So essentially, the HAB model, you saw that specific life stages, processes, together with particular environmental conditions, affect the bloom initiation and decay. So those are the nice stories. I also wanted to discuss, so that brought me back to some of the things we wanted to do but didn't work out. And I think those are also provide good lessons and actually give understanding of gaps in the scientific in the things that we were trying to do. So what I learned from things that did not work. So first, that sardine model, that was supposed to be a full life cycle model. That was coupled to a lower trophic or phytoplankton, zooplankton model. Never happened. So this is the dream dream schematic for that full life cycle model, which includes movement, capacity for movement of the sardines, the growth. So not just the larvae, but all the way to the adults spawning and so on. That whole life cycle. We were supposed to use what's called a bioenergetic model to represent the sardines. So they would there's this how much they consume, how much including respiration, metabolic rates, and so on. But so what happened? Why didn't we do it? Because there wasn't enough data. There wasn't enough information to parameterize the Sardinella lemuro life cycle model in a way that we were comfortable enough with, because the example parameters processes were all from more the temperate areas, and it's really difficult to use those for a tropical setting. So what we opted for was what you saw: simplify it, but still make it be meaningful, um, and it still give us insights. But I think, this really illustrated the gaps that we still had about the biology of the sardines here. The other was, for the pyridinium. No, we wanted to look at pyridinium, but there was another project around the same time I was involved in, and we wanted actually to represent plankton functional types in target Philippine coastal waters. So when I talk about plankton functional types, it's we're representing plankton based on some ecological features, like life history events, distinct set of physiological environmental requirements that affects its biomass and productivity. So this is an example from Sweden, where you have, this is nutrient availability, and this is for light, diminishing light. And the idea is that, oh, sorry, mali. I think I made oh I deleted the wrong figure. It should be the other figure. But anyway, the numbers here, they're representing different types of plankton, phytoplankton, and some of those, I think 4 or 5, some are hab species. And so we wanted to embed in a full ecosystem model, like representation of the community of your phytoplankton, and then particular hab types, and assess how they would come out as more dominant in particular conditions, and so on. So what happened was, this was ice cream days. I don't know, Annette, do you remember? Dr. Terry Alino as the program lead at the time. Ice cream. It was on climate change. Were you there? Yeah. But eventually became resiliencies, because I didn't want to talk about climate change anyway at that time. So um, during the point, we tried to figure out how to represent the plankton functional types here in the Philippines. And again, same problem with the sardines. There wasn't much data. Most of the data were from temperate countries. And it would be really, how would you have an optimum temperature that's like 15 degrees in a tropical setting? Of course, that's not going to work. So we did come up with some presentation of the growth parameters and so on, but it wasn't, it didn't pursue it anymore at the time, because it didn't work. Long story short. So those were the things those would be, some things that we need to fill in. I think we're still here for the tropical environment, for the Philippines, if we want to move forward with these kinds of studies. So then here are just general perspectives going forward. So for the individual-based approach for ecosystem modeling, I think they're generally very good for species-specific representation. That's easy because, individuals and species representation is easy, and really teasing out detailed mechanisms and processes, linking or tracing traits and life histories to larger bloom or other patterns. Some of the challenges that remain is how to integrate this with ecosystem models and the rest of the plankton community. And this is actually an active field of research until now. And how do I'll talk about trait-based representation, how to include various trade-offs and life history representation and trade-offs into these kinds of individual-based models. So, and then, of course, even more so, how do you represent adaptations and evolutionary scale changes, which makes things more complex? So we have to just keep on making things more complex just because we want it to be we want to represent the complexity but in a meaningful but still tractable way is the point. So hopefully, some work that we're gonna continue through. A new finally going to start after so long. Sustain program where we will be building ecosystem models, and we have to kind of standardize more this method of developing these ecosystem models for different areas in the Philippines. And it is part of what's called the Global Coastal Ocean Experiment. So we will be doing it together with other countries or other areas around the world as part of the UN Ocean Decade Program. And no, this is not a priority in that UN Ocean Decade Program, Coast Predict. Um, I am working also with other colleagues in Jamstec and others to really incorporate more the trait-based representation in these kinds of ecosystem models. So I will not lecture about trait-based representation here anymore. If you're interested in it, we can discuss another time. And the other aspects I want to emphasize, of course, all of that, these things are presented. Collaboration is key. I've been collaborating with many people across discipline, across sectors. So, um, and these studies have come out because of that, not just me, myself, and I. uh, I also those studies I'm presenting the model, but behind the scenes of those models, there have been a lot of laboratory and field work as well, whether it's done by us or from older studies or other labs. So, for example, the toxin parameters have been done by assessing doing lab work for the shellfish and so on. Culture experiments for all those parameters and the growth of pyridinium. So those very critical parts. And I think it's just if you have a framework of the whole system, it makes it easy to assess, okay, we need this data, but those aren't available right now. And so if we want to assess what's, the important things, then we need to do these experiments or the lab field and so on. And of course, the lab has changed a lot through the years and also has maintained people through the years. Good and bad. And, so we're a diverse group, I think, and but yeah, I think we contribute to these kinds of frameworks in our own ways. So that's it. So thank you very much.
Okay, maraming salamat, Alet. So now we'll open the floor to questions. Siguro we'll start with the people here, especially the students. Any questions, comments? Wala. Ano ba to, Alet? Okay, go. Collaborative effort. Okay.
Alat, can you walk us through the specific mga parameters that experimentalists need to measure in order for the models to happen? So, um,
Ito ba na? Palitan mo.
the earlier model we're talking about to make phytoplankton groups
Oh.
are the parameters that we need to measure, right, to feed into the model.
Species specific stuff. So for example, your Alexandria model, or then that would be typically your growth with under different light, temperature, nutrient conditions, and then well, ideally, you have mortality rates as well, and grazing rates. But then, yun nga, I mean, if you don't have all of those, then probably you won't have all of those. And of course, there's a certain, there's the applicability of the study to to a wider range of conditions. You can there's ways naman to what we call calibrate. So that's why it's also important to have field data, because if you have the actual data of a bloom, the abundance of your pyridinium through time, then you can match the model outcome, even if you don't have all the parameters that you need, right? So both are important. I don't know. Or do you want others? I can give you a list. There's a long list.
Is that okay, Adi, for now?
Yeah.
MJ? Anybody else? Somebody looking at the Zoom? Question? Meron bang? Wala pa rin questions. Yes.
Hello po, good afternoon. I'm Mika from Oco Lab. So, bago pa lang po ako sa MSI. And thanks for your super interesting presentation. So, remote sensing naman po yung background ko. And I was wondering, kasi you mentioned yung big gap na input yung phytoplankton functional types. And I was wondering, given the advent of hyperspectral data, where you could potentially distinguish the different, let's say, spectral signatures of different phytoplankton. Sa tingin niyo po, magiging helpful po ba yun? Kasi, yeah, first off, but sa tingin niyo kaya ma-distinguish yung different groups potentially, and also in terms of signal from satellite data. Kasi minsan, toxic na yung algal bloom at lower concentration. So, based on your experience or, yeah, may ano po ba, kahit smaller concentrations, may pag-asa bang makita or ma-detect from satellite data? So, yeah.
Okay, thanks. Yeah, good question. So for the first part, actually even before, not even the hyperspectral, but even just the non-hyperspectral ones, they have some people have been using it already to distinguish your functional types and even specific species. So, for example, in Florida, they use it to detect Carenia, and also I think in Japan, some other species. So yes, it's possible, but that's really it's better for the ones with a high biomass species. For the ones that are low biomass blooms like pyridinium, toxic, alexandrium, yeah, it's more difficult because even if like what you said, even if there's only a lower cell concentration, even tens of cells to hundreds of cells per liter, that can also already be toxic and that can't be detected well by the satellite. And then of course your problem here in the tropics with the clouds. So the time when a lot of blooms are occurring, which is southwest monsoon, is the time when it's cloudy. So better, I would love about this. It's better to have a drone and that's below the crowd. So then you have your hyperspectral sensor. Yun.
Thank you.
Thank you. Okay, so yun na yung next gen. Actually, meron ng potentials for drones to do that, no? Parang ala LiDAR na hindi na eroplano. Drone na. Yes, Mike.
Very interesting presentation. Let mo those are sardines because you raised the question whether having a closure tapos letting the sardines spawn is beneficial to the fisheries or not. But I guess you have shown it that it's indeed beneficial. It's counterintuitive. For example, if you look at your chlorophyll, which is very high, and then you would expect more larvae to be there because of the availability of food. But then your data at first showed that there was low retention, as you said, low retention. I wonder if that's low retention or that's you're seeing a residual from predation because you had prey number, which is constant, and then you increase the number of larvae from the closure, and then you increase now the succeeding years, no succeeding period, you're actually seeing an increase in the number of larvae. So that means that the predator has a very, the presence of the prey, and the pulse that you see there is actually the impact of predation rather than physical processes that leads to the retention of the larvae. Nandun sa result mo sa sardines. That's why I related to the benefit of closure. Kasi marami ka ng larvae ngayon dahil marami nang nangitlog. And hindi sa predation mo. Kaya nakita mo doon sa tumaas yung number of larvae mo. And then you interpret it as a retention. Yung physical processes hindi naman nag-change eh. The only thing that changes is the number of
This one?
larvae. Oo.
This one?
Yeah, oo. Ganyan.
So, uh, so well, wait, sorry, ano mo sa, actually, this is chlorophyll, potential prey, food of the larvae.
Yeah.
Ang ano lang namin dito is actually, I mean, in general, it's high. And it's based on other studies, it's good enough to support, it's the typical upwelling productive areas, yung level ng chlorophyll niya. But wala naman kaming pinasok na walang predation dun sa sardine larvae
Yeah.
itself. So, the only way in the model that it can be lost is actually through yung hindi siya ma-retain circulation, mawawala siya from that the domain in the
So,
area.
yun yung interesting thing na titingnan, yung impact of the predation. Kasi nga nag-closure ka eh. Tapos all things are the same except for the number of larvae na you increased. And then you interpret it as retention. When in fact it can also be influence of predation. Dahil there's safety in numbers. Marami sila masyado. Hindi na sila nakapos ng predation ngayon. Dumami sila kasi dumami yung input mo eh. Dumami yung ano mo eh. Larvae. So yan lang.
I think yun yung mga
Related to the fisheries aspect.
yun yung pwedeng mas ma-tease out kung full cycle na sardine model with the predation on the.
Okay, so we have a question sa Zoom.
Hi, ma'am. We have a question on Zoom from Lawrence Patrick Bernardo. Do you have any plan? Do you have any plan to explore or introduce machine learning / artificial intelligence approaches in your modeling frameworks?
Hi, Lori, from Japan. Yes. So actually, well, in terms of yung sustain program, where one step is we collaborate. The idea is to use AI so that if we develop the models, it will develop at least some of the aspects, the parameterization or the configuration of the model using AI para mas madali sa buhay namin. So that's one in terms of the physical configuration. In terms of the more biological aspect, yeah, I mean, I haven't really thought about it in that light for these mechanistic models because the one I'm thinking more of is how to represent the traits. But that actually, now thinking about it like that, we can actually maybe use AI to if we have if we have good databases to get those traits and to use those to come up with those model representations. But we'll see.
Another question po on Zoom from Richard Reddill. Good day. Thank you for your talk, Dr. Allet. With the increasing data we have in literature regarding nutrient requirements of HABs, and some of this literature is contrasting, how would we distill these before inputting them into a model? How would data from lab experiments translate into models as compared to field-generated data?
Both but whether it's lab or field, of course, there's always limitations. Yeah. So lab is you have your more limited conditions and usually one, two factors only. So we're not talking about synergistic outcomes there. So the it is more of consensus based. So based on more, if we summarize, synthesize studies across all of these lab experiments, across all these field experiments, what can we say? And can we then that's use that for the model representation processes. And things start. Yes, of course. Mm. Mm. Mm.
Now, also on the division of the cells.
Yes.
So which
Yeah.
is a measure, reduction, yun yung measure ng increase in the growth of the
Mhm.
population. Kakailangan din yun tignan. It's just nutrient, it's not a static thing.
Mm.
It's sure. No? For example, the changes within the yield, halimbawa. Diba? So we know that these animals reproduce so fast. So the fine scales are very important. No? Hindi siya months, weeks.
Yeah.
It's a few hours or within
Yeah.
the day. No?
Yeah.
So, and I don't know if many of the phytoplankton, diba, they usually know when there's light, no? So baka kailangan ipasok yung some of those things in terms of their inherent
Yeah.
productivity in terms of cell division, in addition to the cell. Kasi kung nutrient range lang, they're very fine scale kasi itong phytoplankton. So, it's something for even the culture-based work. We need temperature control that you can regulate sa umaga, ganito. Kasi the temperature within the day, no, aside from the light, no?
Yeah.
Paano ba yung cycle nila, di ba? Yung photosynthetic cycle, may diel, ano yan eh.
Yeah. So I mean, obviously these are used for global models as well. So the thing is, in my perspective on it depends on what scale question you're asking the model, right? If you're talking about global or large scale models, it would be very difficult to represent the finer processes like that. So typically you assume that okay, you do a daily calculation and that's enough to represent because you were interested more in months, years, and so on. So but if you're interested of course in the more specific life history processes and how that impacts particular environments, then I think then you could it would be more important to incorporate that.
Okay, so meron pa ba comments? So I hope this is for our students, no? Think about processes in the water column, very important siya, no? Very crucial, especially of those who are very interested in pelagic systems, no? Then of course it's the benthic pelagic interaction I have to pitch
Yeah.
because that's very, very important, no? And here you have your cells, but yun nga, pero there's more to that. A lot of the animals also in the benthos are affected. So kung wala na, okay, so we'd like to thank Aletta for a wonderful talk. Thank you very much, Aletta. And we look forward to the next seminar series for the 50th. Hindi ko alam. Oh, anyway, we'll you wait for the announcement of the next seminar. So maraming salamat.
Thank you everyone.
We have snacks po sa
Pamilya mo ata,
cafeteria,
no?
donut and coffee
Okay.
and
Hi,
chocolate.
thank you for viewing in and people in the Zoom. Welcome. Oops.